vocabulary and some basic principles that you need to understand in order to watch the other videos on genetic diseases. So let's start with some basic terminology. The genome is the DNA contained in the nucleus of a eukaryotic cell. It's considered the hereditary material that can be passed on to successive generations of cells. Genes are portions of the DNA or genome inside a nucleus of a cell, and they code for proteins that carry out specific functions. A chromosome is a rod-shaped cellular organelle. You can see these under the microscope. I'll show you a picture of chromosomes in a minute. And a chromosome contains a single continuous DNA double helix strand, which contains a collection of our genes or DNA. And human cells have 46 chromosomes arranged in 23 pairs. The first 22 chromosomes are numbered 1 through 22, and then we have the sex chromosomes X and Y. And there are 2 copies of each chromosome 1 through 22. These are called a homologous pair. And a key point here that you need to understand in order to understand genetics is that in general, we have 2 copies of most of our genes on a chromosome. This is a karyogram, which is a picture of the chromosomes inside a human cell. And you can see that there are 23 pairs as we just discussed. Most of the cells in the human body are somatic cells. These cells are referred to as diploid, meaning they contain 2 sets of chromosomes in 23 pairs like we just saw in the last picture. A small subset of our cells, however, are called gametes. These are reproductive cells, and they are haploid. They have only 1 set of chromosomes. They're designed to merge with another haploid cell to create a diploid offspring. The somatic cells in our body replicate through a process called mitosis. This involves an S phase of the cell cycle where the chromosomes are replicated into 2 sister chromatids. Then there is an M phase, which is where mitosis occurs, where the cell divides, and a daughter cell is created with copies of the chromosome. To understand this, I've drawn a picture at the bottom of the screen. So we begin with a somatic cell, which has a homologous chromosome pair, 2 copies of a given chromosome. For example, let's say this is chromosome number 11. In the S phase of the cell cycle, DNA is replicated and the chromosome becomes 2 sister chromatids, as shown in this picture here. The cell then divides to become 2 daughter cells, which contain copies of the original chromosomes. Reproductive cells undergo a process different from mitosis. It's called meiosis. This occurs in gametes. This generates cells that are haploid with 1 set of chromosomes. It's generated by the process of meiosis of cells called germline cells. And then 2 haploid cells will merge in fertilization to create a new diploid cells with half of the chromosomes coming from one parent and half from another. So a key point here, which is another thing that comes up again and again in genetics, is that in general, offspring get 1 gene from their mother and 1 from their father. Some more terminology on this slide. An allele is an alternative form of a gene. It turns out that many genes have several forms. For example, the gene for eye color has multiple different forms, and that's why human beings have different eye colors. The same is true of many other genes. And in genetic textbooks and in genetic descriptions, this is often represented by a letter. For example, the A gene will have 1 form denoted by a capital A and another form denoted by a lowercase a. When we say a gene displays genetic polymorphism, we mean that it exists in multiple forms and has many different alleles. When we discuss the locus of a gene, the plural is loci. We're talking about the location of the allele on a chromosome. That's what the word locus means. So don't get confused. Remember that our cells contain DNA, which makes up our genes. Genes are found in multiple different forms called alleles. Each allele is located at a particular spot on a chromosome called a locus, and you can find the loci for various genes on particular chromosomes. The genotype is the genetic makeup of a cell or individual, and it often refers to the names of 2 copies of a gene. For example, if a person inherited gene A from their father and gene B from their mother, that person's genotype would be said to be AB. Or sometimes it refers to 2 alleles of gene A. For example, if gene A comes in the form big A and the little a form, the person's genotype might be 2 big A's, 1 big A and 1 little a, or 2 little a's. The phenotype, on the other hand, is the physical characteristics that result from a genotype. For example, genotype AB might equal phenotype blue eyes, and genotype BB might equal phenotype green eyes. The wild type version of a gene or allele is the type that's common in most individuals. For example, for many genes that come in more than 1 allele, they will say the capital letter version of the gene, such as capital A, is the wild type. The mutant version of a gene or allele is different from the wild type. It's caused by a mutation, and for example, the lowercase letter often denotes the mutant in genetic examples. So an individual might have genotype capital AA, meaning they have two wild type or normal versions of the gene, or they might be a mixture with one capital A and one lowercase a, or they might have two copies of the mutant version of the allele. When we say a cell or an individual is homozygous, we mean that they have two identical copies of a gene. For example, imagine the A gene comes in two alleles. One we'll call capital A, the other we'll call lowercase a. A homozygous individual could have two copies of the capital A or two copies of the lower a version. A heterozygous individual or cell, on the other hand, has two different copies of the gene, such as capital A and little a. Mutations in our gene are what lead to different alleles and different versions of a particular gene. So let's talk about types of mutations. A germline mutation is a mutation that occurs in the DNA of sperm or egg cells, in other words, of gametes. This type of mutation can be transmitted to offspring, and when it is, the DNA mutation will be found in every cell in the body of that offspring that inherited that germline mutation. Somatic mutations, on the other hand, are acquired during the lifespan of a somatic cell. They are not transmitted to offspring. So, for example, if one of your skin cells acquires a mutation, that is never going to be passed on to your offspring. However, it will exist in all the daughter cells of that particular skin cell. A dominant version of a gene or a dominant allele is one that can determine the phenotype even among individuals who have only a single copy. It is often denoted with capital letters. For example, if a given gene has two alleles and one is denoted by capital A and the other by lowercase a, then individuals who have one copy of capital A and one lowercase a, or individuals who have two copies of capital A, will all have the A phenotype. In other words, the capital A allele dominates over the lowercase a allele. In contrast, a recessive gene or allele requires two copies to produce a phenotype. This is often denoted with lowercase letters. For example, only individuals who have two copies of the lowercase a allele will have the lowercase a phenotype. Individuals who have a capital A and lowercase a, or two capital A's, will have the capital A phenotype. Codominance occurs when there are different alleles for a given gene, but none are dominant over the other, and both alleles in an individual contribute to the phenotype. The classic example of codominance is the ABO blood groups. If you have the A gene, then your red cells will have the A antigen on their surface. If you have the B gene, then your red cells will have the B antigen on their surface. And if you have the O gene, your red cells will have neither A nor B. Individuals who are A and B express both the A and B antigens. That is because they are codominant. Neither one dominates over the other and excludes the effects of the other. The rare condition alpha-1 antitrypsin deficiency also displays codominance, and this is often tested on step 1. This condition may cause early COPD and liver disease, and I discussed the clinical aspects in detail in the video in the pulmonary section on obstructive lung disease. But for our purposes here, just know that mutations in the AAT gene, which produces alpha-1 antitrypsin deficiency, lead to this disorder. The M allele is the normal version of the AAT gene. If you have the S allele, you will produce moderately low levels of the alpha-1 antitrypsin protein. If you have the Z allele, you will produce severely reduced protein levels of alpha-1 antitrypsin. So the combination of alleles determines protein levels, and this is because they display codominance. So if an individual is MM, that individual will be normal. If an individual is ZZ, that individual will have a severe deficiency of alpha-1 antitrypsin. Other combinations lead to variable levels of disease depending on how much alpha-1 antitrypsin protein is produced. Penetrance is a genetic term used to describe the proportion of individuals with an allele that express the phenotype for that allele. And there are a number of diseases which display what's known as incomplete penetrance. This means that not all individuals with the disease mutation develop disease. It's often applied to autosomal dominant disorders where you would expect everyone that carries the gene to have disease. However, in disorders that have incomplete penetrance, not all patients with the autosomal dominant disease gene develop disease. And a classic example of genetic mutations that display incomplete penetrance are the BRCA1 and BRCA2 gene mutations. BRCA1 and BRCA2 are genetic mutations that lead to breast cancer. They are germline gene mutations and they are autosomal dominant. However, many women born with these mutations do not develop cancer, and this has important implications because although women that have these gene mutations are at increased risk, they may not develop breast cancer. And therefore, if you are going to apply a cancer risk reduction strategy like prophylactic breast surgery, you don't know whether or not it'll benefit the patient since they may not develop cancer because these gene mutations display incomplete penetrance. Expressivity refers to variations in phenotype of an abnormal gene. This is different from penetrance, and this is often a point of confusion. To understand this, let's discuss a classic disorder that displays variable expressivity, and that is neurofibromatosis type 1. This is a neurocutaneous disorder where patients develop brain tumors and characteristic skin findings. It's an autosomal dominant disorder, so you expect all individuals to have disease, and in fact, they do all have disease. This disorder has 100% penetrance. However, there's great variability in the disease severity, the number of tumors and the skin findings, and for this reason, neurofibromatosis has variable expressivity, although it has 100% penetrance because everyone with the gene does get the disease. Pleiotropy is a term that refers to the fact that one gene or one gene mutation can have multiple phenotypic effects and traits. For example, a single gene mutation could affect the skin, the brains, and the eyes. And there are many examples of disorders which are caused by a single gene mutation, yet lead to findings in multiple different organ systems. For example, phenylketonuria or PKU can affect the skin. It leads to a characteristic body odor, and it also causes mental disability. Marfan syndrome leads to findings in the limbs, the eyes, and the blood vessels. Cystic fibrosis can involve the lungs and the pancreas, and osteogenesis imperfecta can involve the bones, eyes, and hearings. And there are many other examples of genes that have pleiotropic effects and genetic disorders that are caused by a single gene mutation but lead to abnormalities in multiple organ systems. For the next few slides, I'm going to discuss the two-hit origin of cancer. This refers to mutations in tumor suppressor genes that can lead to cancer. And the word tumor suppressor gene is very important because the two-hit origin of cancer only applies to mutations in these genes. Tumor suppressor genes are genes with many roles in cells. They are often gatekeepers that regulate cell cycle progression. Sometimes they are DNA repair genes. And if you have a heterozygous mutation for one of your two genes of a tumor suppressor gene, this will lead to no disease. You need to have mutations of both alleles to develop cancer. And for this reason, it's said that cancer requires two hits, meaning an abnormality in both copies of a tumor suppressor gene, or another way this is sometimes stated in genetic textbooks, is that you need loss of heterozygosity in order to develop cancer related to a mutation of a tumor suppressor gene. Here's a picture from the web just to make this very clear. A normal individual has two copies of tumor suppressor genes, one found on each copy of a homologous chromosome pair. If they develop a mutation and lose one of the tumor suppressor genes, they still won't get cancer because the other gene will continue to function. This is said to be the first hit. However, if they develop a second hit and lose the other copy of the tumor suppressor gene, they will then have unregulated cell growth in that particular cell that has lost both copies of the tumor suppressor gene, and that will lead to no functional tumor suppressor effect, and that will lead to malignancy. The classic example of a tumor that illustrates the two-hit origin is retinoblastoma. This is the tumor in which the two-hit origin of cancer was originally described. It is a rare childhood eye malignancy, and 40% of cases are hereditary. In these cases, children inherit one mutated gene that's found in all cells of their body at birth. This is an example of a germline mutation. They then, at a later point in childhood, get a second somatic hit that takes out the second copy of the tumor suppressor gene. Because they are born with one abnormal form of the tumor suppressor gene, cancer requires only one somatic mutation in order to develop. Therefore, these children develop frequent and multiple tumors, and they develop tumors at a younger age. Contrast that with the sporadic form of retinoblastoma, which is non-familial. Children who develop this form of retinoblastoma need to have two somatic hits that take out both copies of their tumor suppressor genes, and two mutations in the same cell is a rare occurrence. Therefore, these children do not develop multiple tumors, they develop a single tumor, and in addition, the tumors usually occur at a later age. Another example of the two-hit origin of cancer is HNPCC or Lynch syndrome. HNPCC stands for Hereditary Non-Polyposis Colorectal Cancer Syndrome, and I discussed this in detail in the GI section, but for our purposes here, you should just know that this is an inherited colorectal cancer syndrome that involves a germline mutation in DNA mismatch repair genes, and these mismatch repair genes are another example of a tumor suppressor gene. So in this disorder, you have a parent who passes on two copies of a tumor suppressor gene to a child, one of those copies is abnormal because of a germline mutation. This means that gene will be abnormal in all the cells of the offspring's body. Eventually, the offspring will develop a second mutation in one of their cells, and this will lead to cancer. In addition, because that offspring has that mutation in all cells of their body, it will also be present in their germ cells, and therefore they can pass it on to the next family member. And therefore, this tends to be a family which has lots of colorectal cancer, all because of this germline mutation. Another example that also involves the GI tract is familial adenomatous polyposis, or FAP. This is a germline mutation in the APC gene, another tumor suppressor gene. It always, 100% of the time, progresses to colon cancer. In fact, the risk of colon cancer is so high that people born with this germline mutation have a colon removal or a colectomy surgery prophylactically to prevent the development of colon cancer. And then one final example is the Li-Fraumeni syndrome. This is a syndrome of multiple malignancies that occur at an early age. It's sometimes called the SBLA cancer syndrome. SBLA stands for sarcoma, breast, leukemia, and adrenal gland. These are all places where tumors can develop. This is caused by another germline mutation, but this time in a tumor suppressor gene called TP53. This codes for tumor protein P53, which delays cell cycle progression to allow time for DNA repair. So once again, people inherit one copy of the TP53 gene that is abnormal. Over the course of their lives, in some cells in their body, the other copy becomes abnormal, and that leads to cancer. Now let's talk about mosaicism. This is a genetic term that refers to gene differences in cells of the same individual. So let's imagine that we have a whole bunch of blue cells which are replicating to create daughter cells, and one of them develops a mutation during the course of mitosis. This will lead to a new population of cells which has different genes compared to the other cells in that same individual. Therefore, develops a mutation differences in cells of the same individual. So let's imagine that we have a whole bunch of blue cells which are replicating to create daughter cells, and one of them develops a mutation during the course of mitosis. This will lead to a new population of cells which has different genes compared to the other cells in that same individual. Therefore, this individual has a mixture or a mosaic of different genetic makeup, and therefore this is said to be an example of mosaicism. Mosaicism can involve germline or somatic cells. Germline mosaicism can be passed on to offspring. Let's imagine that we have a parent who has 3 different germline cells that are producing gametes to form offspring. And let's imagine that one of those germline cells develops a mutation. This is an example of germline mosaicism, and any gametes formed from this mutated cell will have a mutation that can be passed on to offspring. Pure germline mosaicism is very difficult to detect. It's not present in blood or tissue samples that are often used for genetic analysis. What this means is that offspring can develop disease that may appear to be a sporadic mutation, but in fact, it's due to a germline mosaicism in the parent. So this can sometimes present in families as recurrent sporadic cases of disease in the offspring. What's really happening is that a parent has germline mosaicism that they are passing on to the offspring. In contrast, somatic mosaicism refers to gene differences in tissues or organs of an individual that do not involve the germline cells. For example, some cases of Turner syndrome involve mosaicism. Women with this version of Turner syndrome have some cells that are 45X and are missing an X chromosome, where other cells are normal and have 2 copies of the X chromosome. These tend to be milder variations of Turner syndrome. In addition, there are rare forms of Down syndrome that involve mosaicism. In babies born with these variants of Down syndrome, some cells have trisomy 21, which leads to Down syndrome, but other cells have 2 copies of chromosome 21, which is normal. There is a rare genetic disorder called McCune-Albright syndrome that is often used to illustrate the principle of mosaicism. This is a genetic disorder that affects many different systems. In particular, it affects many endocrine organs. One of its characteristic features is precocious puberty that can start at a very early age. For example, females may menstruate as early as 2 years old if they have McCune-Albright syndrome. In addition, there is fibrous growth that occurs in the bones. This leads to fractures and bony deformity. And then finally, there are characteristic skin findings. The skin becomes pigmented. These children can develop cafe au lait spots, and the skin pigmentation often has irregular borders that is classically described as appearing to look like the coast of Maine. Shown on the right side of the screen are two children with skin lesions from McCune-Albright syndrome. You can see how this child on the left has skin pigmentation with very irregular borders. And in case you don't know what the coast of Maine looks like, this is a map picture of Maine, and you can see how the coast is very irregular, just like this child's skin lesion on her chest. McCune-Albright syndrome is caused by a sporadic mutation that occurs during development of the child. It is not an inherited genetic abnormality. The mutation that occurs is a somatic mutation of the GNAS gene. This codes for the alpha subunit of a G3 protein that activates adenyl cyclase. And when this gene is abnormal, it leads to continued stimulation of cyclic AMP signaling more than is normal. The most important thing for you to remember about this very rare disorder for the Step 1 exam is what's shown on this slide. McCune-Albright syndrome is caused by a post-zygotic genetic mutation. It occurs after fertilization, and therefore only some tissues and organs are affected. In other words, it is said to display mosaicism, and the clinical phenotype will vary depending on which tissues are affected. So to understand this, just imagine that you have a developing fetus, and it's got a number of different cells here, and one of them develops this mutation. All the daughter cells of this mutated cell will have the McCune-Albright syndrome genetic mutation, but the daughter cells of these other cells will not have the mutation, and therefore the child will develop as a mosaic of genetically modified cells, some being normal, some having the mutation. If there happens to be a germline mutation that leads to the McCune-Albright genetic mutation in all of the cells, that is a lethal condition. In that case, the entire body is affected. So in other words, cells with this mutation can only survive if they are mixed with normal cells in the individual. What this means is if you see a child in clinical practice with McCune-Albright syndrome, that child must have mosaicism. If the child did not, they would have died. They would not have survived to come in for clinical evaluation. I'll finish this video with a couple more terms that you should be familiar with. The first one is genetic heterogeneity. This refers to when the same phenotype develops from different genes or mutations. For example, different mutations of the same allele can lead to the same disease. In other words, there's not a single mutation that leads to the disease. Multiple different types of mutations can cause the disease. We'll talk more about that in a minute. A second example is when different genetic mutations at different loci on the chromosomes cause the same disease. This also is an example of genetic heterogeneity. So the point here is that for many genetic diseases, multiple gene mutations often cause the same disease. Or another way to look at it is that many clinical diseases have multiple genotypes that lead to those disorders. And this is referred to as genetic heterogeneity. One subtype of genetic heterogeneity is allelic heterogeneity. So by way of review, an allele is an alternative form of a gene. So imagine that allele 1 is formed by mutation X of a particular gene, and allele 2 is formed by mutation Y of that same gene. But both X and Y cause the same disease, and X and Y are found at the same chromosomal location or position. Therefore, they involve mutations of the same gene. This is termed allelic heterogeneity. It means different alleles or different mutations lead to the same disease. And the point here is that many alleles possess multiple mutant forms that all lead to the same clinical presentation or clinical disorder. So I've summarized this at the bottom. You have one disease caused by multiple genes, but all those genes are variants of a gene at the same location. Therefore, they are allelic variants, and therefore this is termed allelic heterogeneity. A couple examples of allelic heterogeneity are shown on this slide. Beta thalassemia, which I discussed in the hematology section, involves mutations of the beta globin gene, and there's a wide spectrum of disease severity depending on the mutation. People that are said to possess the beta 0 form of the gene have no function. People who possess the beta 1 form have some function. And I talk about this in more detail in the hematology section. Cystic fibrosis is another example of a disease that displays allelic heterogeneity. Cystic fibrosis is caused from mutations of the CFTR gene, and there are over 1,400 different mutations that have been described that can lead to various forms of cystic fibrosis. Another subtype of genetic heterogeneity is locus heterogeneity. This refers to when mutations in different loci on chromosomes cause the same phenotype. So imagine that we have a chromosome, say it's chromosome number 1 here, and a mutation at this gene right here causes a disease. And then over here we have chromosome number 3, and a mutation here causes the same disease. This is the same disease coming from genetic mutations in 2 different loci on chromosomes, and this is an example of locus heterogeneity. And one example of this in clinical practice is retinitis pigmentosa. This is a rare disorder that causes visual impairment, and there are many different forms of this disorder. There's an autosomal dominant form, a recessive form, an X-linked form, It turns out that mutations at 43 different loci on chromosomes can all lead to this disease. Therefore, retinitis pigmentosa is said to display locus heterogeneity. And I've summarized the key points at the bottom. In locus heterogeneity, we have one disease caused by multiple genes at multiple different locations on chromosomes. And that concludes our video on genetic principles. Hi, everybody, and welcome to our module on genetic mapping. Let's start by reviewing the principle of genetic recombination. During meiosis, chromosomes exchange segments so that the child inherits a patchwork of parental chromosomes and never an exact copy of one of the parental chromosomes. To understand this, let's imagine we have a father and mother with two copies of a chromosome. For example, let's assume this is chromosome number 15 here, and the father has two copies and the mother has two copies. Those chromosomes are going to recombine during meiosis such that child 1 inherits one copy of chromosome 15 from his father and one from his mother. But that chromosome 15 from the father is a mixture of the two chromosome 15s that were present in the father's cells, and the same is true of the mother. And this mixing of genes is called genetic recombination. The next concept we need to explore is what's known as independent assortment. To understand this concept, let's suppose a father has two alleles of his F gene and two alleles of his M gene. Let's suppose that one of his F genes is in a form that we'll call capital F, and the other F gene has a different form, which we'll denote as lowercase f. Let's also suppose that he has two versions of the M gene, one is capital M and one is lowercase m. Let's further suppose that F and M are found on different chromosomes. If this is the case, the F and M genes will independently assort. This means that they can independently recombine, and we'll get a 25% chance of each mixture of the F and M genes in a gamete. To make this more clear, let's look at this picture I've drawn on the screen. So we have a father who has chromosomes within the cells of his body. On chromosome 1, he has the F gene, and one of his copies of chromosome 1 has the capital F gene. The other copy of chromosome 1 contains the lowercase f version of the F gene. Similarly, on the father's chromosome 2, he has two copies of the M gene. One is capital M, the other is lowercase m. Because these genes are found on different chromosomes, they will independently assort, and the gametes will be a mixture of all the possibilities. In other words, 25% of the gametes will contain a capital F and a capital M gene, 25% will contain a lowercase f and a lowercase m gene, and then 25% will contain mixtures of a capital and lowercase f or m. Now, in that example, M gene, different F version of the F gene. Similarly, on the father's chromosome 2, he has two copies of the M gene. One is a capital M, the other is lowercase m. Because these genes are found on different chromosomes, they will independently assort, and the gametes will be a mixture of all the possibilities. In other words, 25% of the gametes will contain a capital F and a capital M gene, 25% will contain a lowercase f and a lowercase m gene, and then 25% will contain mixtures of a capital and lowercase f or m. Now, in that example, the F and M genes were able to independently assort because they were found on different chromosomes. But now let's consider what would happen if the F and M genes were found on the same chromosome. To understand this, let's look at the example I've made on the screen here. Let's assume we have a parent with two copies of chromosome 1 found in the cells of their body. Each copy of chromosome 1 has an F and an M gene. On one version of chromosome 1, there is a capital F and capital M gene. On the other version of chromosome 1, there is a lowercase f and lowercase m gene. What would happen if this parent were to generate gametes? What combinations of the alleles of the F and M gene would they have? Well, it turns out if the F and M genes are very far apart on chromosome 1, then crossover will occur in meiosis to such a degree that you will get the same mixture of combinations of the F and M alleles as if the F and M genes were found on separate chromosomes. In other words, there's a 25% chance that you would create a gamete with a capital F, capital M version of the F and M genes, a 25% chance that you would create a gamete with a capital F, lowercase m, and also a 25% chance of the two other combinations. And this is what occurs if the F and M genes are very far apart on chromosome 1. In this situation, we say that there is a 50% chance that the gametes will have a parental version of the F and M genes. In other words, there's a 50% chance that a gamete will have either a capital F, capital M gene, just like we found in the parent, or a lowercase f, lowercase m gene, just like we found in the parent. There is also a 50% chance that the gametes will have what's known as recombinant versions of the F and M gene. In other words, a capital F, lowercase m, this is a mixture of F and M genes that was not found in the parent, or a lowercase f, capital M, another mixture not found in the parent. Now let's consider what would happen if the alleles of the two genes are very close together. In this case, there is very little crossover, and there will be a low occurrence of the recombination genes, capital F lowercase m or lowercase f capital M. At the extreme, you would get what I've shown on the screen here. If F and M genes are very, very close together on chromosome 1, you would get 50% of gametes having a capital F and a capital M, just like you had in the parent, a 50% chance of having a little f, little m, just like you had in a parent, and you would have a 0% chance of getting any of the recombinant chromosomes because no recombination is occurring because the genes are so close together on chromosome 1. The reason there is little crossover and recombination when genes are close together is because of what I've shown in the screen here. Imagine that the A and B genes are very close together. Only a break in this small section of DNA is going to allow them to recombine. Now imagine that the B and C genes are very far apart. There is a huge span of DNA in which any break will allow B and C to recombine. Thus, you are going to get lots more recombination of the B and C genes relative to the amount of recombination you will get of the A and B genes. So now we can define an important term known as recombination frequency. This is the frequency of recombined genes. In the example we went through on the last few slides, this would be gametes with a capital F lowercase m or a little f capital M. Recombination frequency is denoted by the Greek letter theta, and it ranges from 0 to 0.5. If we go back to this slide here, in this example, genes F and M were very far apart, and the frequency of recombined gametes was 50%. This would be a theta of 0.5. And on this slide, where we considered what would happen if the F and M genes were close together on the same chromosome, there was no occurrence of the recombined genes in the gametes, and therefore this would be an example of theta equal to 0. And a key point here is that the recombination frequency is proportional to the distance between the two genes. If the genes are close together, theta will be 0. If they are far apart, theta will be 0.5. And this can be used to create a genetic map of our genes. So now we've gone through all the concepts that we need to understand in order to discuss genetic mapping, which is also called linkage mapping. This is done by studying large families and tracking the frequency of genetic recombination and using that frequency to determine relative gene locations. So for example, let's suppose we have 3 genes, A, B, and C. Let's suppose that the frequency of recombination of A and B is 16% in a particular family. Recombination of A and C is 0.08 or 8%, and recombination of C and B is also 0.08 or 8%. This means that A, C, and B must have relative locations on a chromosome like I've shown at the bottom of the screen here. A and B are so far apart that they generate a recombination frequency that's higher of 0.16. A and C and A and B are far apart such that they generate a recombination frequency that is smaller of 0.08. This is called linkage mapping or genetic mapping, and it's used to map our genes. This brings up an important term that we need to discuss, and that is the term linkage. This is the tendency of genes or alleles to transmit together. When alleles are more linked, they display less independent assortment. Genes that are close together that have a theta closer to 0 are said to be tightly linked. Genes that are far apart and have a theta closer to 0.5 are said to be unlinked. I'm going to finish this module by discussing the concept of linkage disequilibrium. This is used to study genes that are very close together. When genes are very close together, recombination events are rare and family studies are impractical. Even if you have a large family, you may never see a recombination event, and therefore you'll never be able to calculate a value for theta. Therefore, the principle of linkage disequilibrium is used to study these types of genes, and this is done by studying large populations, not families. Instead, this is applied to populations of individuals. To understand linkage disequilibrium, let's first talk about linkage equilibrium. And to understand this concept, let's assume there are 2 genes found in a population of individuals. Let's assume gene A exists in 2 polymorphisms. There is a big A allele and a little a allele. Let's assume that big A is found in 50% of individuals in a given population, and little a is found in the other 50%. Let's also assume that gene B has 2 polymorphisms. It can exist as a capital B or lower B allele. Let's assume big B is found in 90% of individuals and little b in the other 10%. And I've listed these numbers at the top of the screen here to remind you what they are. Once we know the percentage of individuals with each of the different alleles of the A and B genes, we can then determine what the population frequencies should be for mixtures of those different alleles. The frequency of a big A and a big B found together should be 0.5 times 0.9, which is 0.45. Similarly, a little a and a big B together should be 0.5 times 0.9, which is 0.45. And we can go ahead and calculate all the expected frequencies of the mixtures of those alleles. And if this were to be what we actually found in the population, we would say that these two genes demonstrate what's known as linkage equilibrium. And just a side note here, don't think that you can apply the Hardy-Weinberg law to these gene frequencies here. The Hardy-Weinberg law is meant to determine the frequency of the same gene with different alleles occurring together. In other words, the frequencies of big A's and little a's occurring together. Here we're talking about two different genes, and in this situation, the Hardy-Weinberg law does not apply. So now that we understand linkage equilibrium, we can talk about linkage disequilibrium. This occurs when the population frequencies are higher or lower than expected. For example, imagine if we found an actual mixture of the big A and big B genes in a population of 0.75. That is much higher than the expected frequency of 0.45. This means that this haplotype AB is in linkage disequilibrium. And genes will display greater degrees of linkage disequilibrium when they are closer together, and thus you can use the degree of linkage disequilibrium in a population to study genes that are very close together, too close together to study using families. To understand this more, let's consider a new gene mutation that we'll denote as capital A. And let's suppose that this gene mutation occurs in a gene that is very close to gene B. Initially, A and B are going to be transmitted together in a population. They are so close together that they will be transmitted together from one generation to the next. Now, over time, A and B may eventually separate. Random chance may eventually create a break just between A and B so that they separate. The likelihood of these two genes separating depends on their distance apart from one another and the size of the population. In other words, how many generations have had the opportunity to split A and B from one another. The degree of linkage disequilibrium will be greatest when a gene first enters a population. In other words, when the mutation first occurs. In that situation, A and B are going to be right next to each other, and you're not going to find them separated from one another on many occasions. With successive generations, the degree of linkage disequilibrium will fade. In other words, as the population grows, you will see less linkage disequilibrium. It will also fade if the distance between A and B is greater. So I've summarized these points on this slide. Linkage disequilibrium is affected by the genetic distance between two genes and the times the alleles have been present in a population. And a key point here is that different populations can display different degrees of linkage disequilibrium. If a gene has moved through a population of people for a longer period of time, in other words, if that was the population that first developed a mutation, they may display a different degree of linkage disequilibrium compared to another population that developed a mutation at a later point in evolution. And that concludes our module on gene mapping. Hello, everyone, and welcome to our module on meiosis. Meiosis is the process by which diploid cells, which have two copies of each chromosome, give rise to haploid cells, which have only one copy of each chromosome, and these cells are called gametes. This process is unique to cells known as germ cells. These are cells like spermatocytes found in males and oocytes found in females. And meiosis is broken down into two steps. The first step is called meiosis 1. The second step is called meiosis 2. What happens in meiosis 1 is that diploid cells become haploid cells. This is a process referred to as reductive division. Diploid cells have two copies of each chromosome, and therefore they are often referred to as 2N. Haploid cells have one copy of each chromosome, and therefore they are often referred to as 1N. In meiosis 1, homologous chromosomes separate. So let's talk about what that means exactly. At the bottom left side of the screen, I've drawn a diploid cell. This cell has 2 copies of every chromosome. However, for simplicity's sake, I've only shown one particular chromosome. Let's imagine that this is chromosome 21. Every one of our chromosomes will go through the process on the screen, but we're going to focus in on it in detail for one particular chromosome. For argument's sake, let's say it's chromosome 21. So this is a diploid cell which has a paired chromosome 21, or in other words, has 2 homologous copies of chromosome 21. One of those copies, denoted by the blue line here, came originally from the father. That's the paternal copy of chromosome 21. The other copy, denoted by the red line, came originally from the mother. During interphase, the 2 chromosomes will be replicated and they will become 2 chromosomes with sister chromatids. This is the paternal copy of chromosome 21. It has 2 sister chromatids attached together. The same is true for the maternal copy. The process of meiosis 1 then begins, and the first thing that occurs is there is some crossover or recombination of genetic material. So the 2 chromosomes, the one originally from the father and the one originally from the mother, exchange some genetic material. They then go through a cell division and become haploid cells that are only 1N. These cells that result from meiosis 1 have a single copy of chromosome 21, and that copy is a mixture of the chromosomes that originally came from the father and the mother. In the second step of meiosis 2, the sister chromatids separate and form 4 daughter cells. So here we've got 2 cells which have sister chromatids, and they separate and create 4 gametes, and all of these cells are 1N, meaning they have only 1 copy of each chromosome. And here's a summary slide of the entire process. You begin with a cell with 2 copies of each chromosome. During meiosis 1, the homologous chromosomes separate and undergo crossover and recombination. During meiosis 2, the sister chromatids separate to create 4 gamete cells that are 1N. So that is how the process of meiosis plays out in general. Now let's talk about exactly how it occurs in the sex organs of males and females. In males, the process of spermatogenesis generates haploid gamete cells. This process begins at puberty. Males have cells called Spermatogonium, which are diploid or 2N. They undergo mitosis, not meiosis, to become cells called primary spermatocytes. These cells are diploid and have both an X and Y chromosomes, just like all the cells in the male. The primary spermatocytes undergo meiosis 1, and they will become cells called secondary spermatocytes, which are now haploid and have only an X or a Y chromosome. Those cells will then go through meiosis 2, and they will become 4 spermatids, which are the cells which contain the separated sister chromatids. They are haploid cells. They contain only an X or a Y chromosome. And then those cells go through spermiogenesis, and they become spermatozoa or sperm, which can then go on to fertilize the egg. In females, the process is different. The generation of gametes is called oogenesis. It begins with primary oocytes, which are formed in utero in the female. These are diploid cells, and they are just beginning the process of meiosis 1, and they are arrested in prophase of meiosis 1 until puberty. And this is a very important point. Meiosis is very drawn out in females, and for that reason, as we will see later, this process is vulnerable to errors that can lead to pathology in the offspring. At puberty, a few of the primary oocytes will complete meiosis 1 each menstrual cycle. Some will form what's known as polar bodies and degenerate. Others will form secondary oocytes, which are haploid and can go on to become eggs. At this point, meiosis 2 begins, but arrests in metaphase. If fertilization occurs, then meiosis 2 will become complete. So once again, the key point here is that this is a very drawn out process in females, and therefore it is more vulnerable to errors than the process in men. Now that we've discussed meiosis, for the rest of this video, I'm going to talk about errors in meiosis that lead to pathology. And let's begin by defining an important term, and that is the term aneuploidy. Aneuploidy means an abnormal chromosome number, either an extra or missing chromosome within a cell. Disomy means 2 copies of a chromosome. This is the normal state. Monosomy means a cell contains only 1 copy of a chromosome. This is a form of aneuploidy. Trisomy is also a form of aneuploidy. This means a cell contains 3 copies of a particular chromosome. Meiotic non-disjunction is a failure of chromosome pairs to separate during meiosis, and it is the most common mechanism of aneuploidy, and it can occur in either meiosis 1 or meiosis 2. If there is a meiosis 1 non-disjunction, the homologous chromosomes will fail to separate. This will generate one cell with no chromosomes, which will go on to generate gametes that have no chromosomes. The other cell will contain 2 copies of a particular chromosome, and it will go to form gametes that have 2 copies of a chromosome. Remember, these gametes are supposed to only have a single copy of each chromosome. They are supposed to be 1N. However, in this case, as you can see, they are 2N. They contain 2 copies of the chromosome that came from the parent. Importantly, note that when there is a meiosis 1 non-disjunction, the gametes have a mixture of genes. In other words, they have 1 copy of the chromosome that came from the father and 1 copy of the chromosome that came from the mother. This is an important feature of a meiosis 1 non-disjunction event, and it is different from what you see in a meiosis 2 non-disjunction. If there is a meiosis 2 non-disjunction, meiosis 1 will occur normally, and the homologous chromosomes will separate. However, in meiosis 2, the sister chromatids will fail to separate. This will generate 1 gamete which has no genes and another gamete which is diploid. But importantly, this diploid gamete has no mixture of the original paternal and maternal genes. Note that in the example I've drawn here, it's got 2 copies of the red chromosome, which initially came from the mother. It does not have 1 copy of the red chromosome and 1 copy of the blue. And this is an important feature of meiosis 2 non-disjunction events. And here's a slide just to pull it all together. If you have a meiosis 1 non-disjunction, the 2 homologous chromosomes will fail to separate. This will generate gametes that are 2N and have a mixture of the paternal and maternal genes from the original individual. If you have a meiosis 2 non-disjunction, the sister chromatids fail to separate. This will create a 2N or diploid gamete that has 2 copies of the same chromosome from the individual in whom the non-disjunction occurs. And you'll note that I've not shown crossover and recombination here, and that's because crossover and recombination is rare in situations where non-disjunction occurs. Monosomy occurs. is when you have fertilization of a 1N or normal gamete with a 0N gamete that contains no copies of a particular chromosome. And we saw how in meiosis there were a couple of ways by which you could generate gametes that did not contain a copy of a particular chromosome. This can lead to monosomy for that particular chromosome. Most monosomies are not viable and will result in a spontaneous abortion. Turner syndrome is one example of a monosomy. It's a monosomy of the sex chromosomes where a girl is born with only one X chromosome. Trisomy occurs when you have fertilization of a normal 1N gamete with a 2N gamete, and we saw a couple of ways by which meiotic non-disjunctions could lead to gametes that contain two copies of a particular chromosome. Most times when there is trisomy, it's not compatible with life and will lead to a spontaneous abortion. However, there are a couple of exceptions. Trisomy 21 is viable and it will lead to Down syndrome, and 95% of cases of Down syndrome are due to meiotic non-disjunction. Trisomy 18 will also lead to Edwards syndrome, which can be viable, and trisomy 13 can lead to Patau syndrome, which also can be viable. Maternal meiosis I non-disjunction errors are a common cause of trisomy. As we discussed earlier in the process of oogenesis, meiosis I is protracted in females. It begins prenatally and it's completed at ovulation many years later. This leaves lots of time during which non-disjunction can occur. In addition, this is the reason why advanced maternal age is a risk factor for many forms of trisomy. With advanced maternal age, the eggs that are being released have spent longer going through meiosis, and therefore there's more time for non-disjunction. When a baby is born with trisomy, the cause of the non-disjunction that led to the trisomy is suggested by the trisomy genotype in the child. To understand this, let's imagine that we have a father with two copies of chromosome 21, and we use genetic techniques to identify unique features of each chromosome 21, and we call one of the father's chromosomes 21A, and the other one we characterize and call 21B. We do the same thing for the mother and characterize her two chromosome 21s as 21C and 21D. Now, if we look at the baby with trisomy, let's suppose the baby has trisomy 21 and has copies of chromosome 21 that are A, C, and D. Well, we know this must have been an error in the mother because there are two copies of chromosome 21 that came from the mother. In addition, we know it must be a meiosis I non-disjunction event because the homologous chromosomes in the mother, C and D, failed to separate. And that's what happens when there's a meiosis I non-disjunction, as we discussed earlier. Conversely, let's suppose that the baby had trisomy 21 and had chromosome 21s A, C, and C. Once again, we would know it is a maternal error because there are two copies of chromosome 21 from the mother. In addition, we would know it's a meiosis II non-disjunction because in meiosis II non-disjunction, the sister chromatids fail to separate and you get two copies of the same chromosome in the fetus. Uniparental disomy is when a child inherits two copies of one parent's chromosome and no copies of the other parent's chromosome. This is a rare event. There have to be abnormal gametes from the male and female. One of them has to contain no copies of a particular chromosome and the other one has to contain two copies. Once again, you can use the genotype of the fetus to figure out where the non-disjunction event occurs. For example, once again, if we have a father with two chromosome 21s A and B and a mother with two chromosome 21s C and D, suppose that the child is born with AA. We know that the child must have uniparental disomy coming from the father because the child has two copies of the father's chromosomes. In addition, we know it must be a meiosis II error because the child inherited two copies of the same chromosome from the father. And this is what happens when there's a meiosis II error. This is called isodisomy. It's uniparental disomy with two copies of one chromosome. Conversely, suppose the child was CD for chromosome 21. This would be a meiosis I error in the mother. This would indicate that the homologous chromosomes in the mother failed to separate, and this is called heterodisomy. A child born with uniparental disomy is euploid, meaning it has a normal number of chromosomes. There is no aneuploidy, and usually it has a normal phenotype. The one problem that can result from uniparental disomy is that it can lead to a phenotype for a recessive disease. For example, suppose the father was capital A, and little a is a version of a gene that is recessive for a disease. If uniparental disomy occurs and the child inherits two copies of the diseased gene, that child will get the disease from a single parent, not in the usual way that recessive disease is inherited, which is from two parents who are carriers. The last topic I'll discuss in this module is that of a Robertsonian translocation. This occurs when there is fusion of the long arms of two chromosomes. This happens to chromosomes that are known as acrocentric. These chromosomes have a centromere very close to their end, and thus they have a very small, short arm. This is true for chromosomes 13, 14, 21, and 22. To understand a Robertsonian translocation, let's look at this picture I've made on the screen. Parent, not in the usual way that recessive diseases in long arms of two chromosomes. This happens to chromosomes that are known as acrocentric. These chromosomes have a centromere very close to their end, and thus they have a very small, short arm. This is true for chromosomes 13, 14, 21, and 22. To understand a Robertsonian translocation, let's look at this picture I've made on the screen. So we have a normal chromosome 14 and a normal chromosome 21. If these two chromosomes undergo a Robertsonian translocation, the long arms will link up and create a fused chromosome 14 and 21. The short arms of both chromosomes will be lost. This usually carries no important consequence for the individual in whom the translocation occurs. It's a small amount of genetic material that is lost, and it usually does not lead to any clinical consequences. The major implication of this translocation is that the person carrying these two fused long arms is likely to have many offspring with chromosomal anomalies. To understand this, let's imagine that we have a normal father who has two copies of chromosome 14 and two copies of chromosome 21. And let's imagine that he mates with a mother who has undergone a Robertsonian translocation. So she has one normal copy of chromosome 14, one normal copy of chromosome 21, and one fused chromosome that resulted from the Robertsonian translocation. At the bottom of the screen, I've shown all the potential zygotes that can result from this union. The father is always going to donate one copy of 14 and 21 because he is normal, but there are many different possibilities for which two chromosomes the mother will donate. If the mother's very lucky, she will donate her two normal chromosomes, 14 and 21, and the child will be normal. If she's a little bit less lucky, she will donate her fused chromosomes, and this will create a child who is a carrier for the Robertsonian translocation. All of the other possibilities are very bad for the fetus. If the mother donates her translocated chromosome and her normal copy of 21, then the baby will have trisomy 21 and will have Down syndrome. And a Robertsonian translocation is one mechanism by which Down syndrome can occur. It's not the most common, but it's one possible mechanism. All of these other possibilities, including monosomy 14, monosomy 21, and trisomy 14, are not going to be viable, and therefore this couple is likely to have many failed pregnancies and many spontaneous abortions. So on this slide, I've summarized the important points. The carrier of a Robertsonian translocation has only 45 chromosomes. One is a translocated chromosome. The carrier has lost the short arms, but they will usually have a normal phenotype because there's no clinical consequence from loss of the short arms and therefore no disease. 13 and 14 and 14 and 21 are the most common, and because 21 is often involved, this is one of the ways you can get Down syndrome or trisomy 21. The main clinical consequences are many monosomy and trisomy gametes, and for this reason, there are frequent spontaneous abortions. The other consequence is that the carrier may have a child with Down syndrome or trisomy 21. Because of the possibility of a Robertsonian translocation leading to fetal loss, a karyotype is a test that's often done in couples who have recurrent fetal losses. A karyotype is used to diagnose chromosomal imbalances. A karyotype creates a karyogram like is shown on the screen here, and you can identify two fused chromosomes from a karyotype. So this is a test that's often sent in the workup of infertility to see if one of the parents is a carrier of a Robertsonian translocation. And that concludes our video on meiosis. Hi, everybody, and welcome to our module on the Hardy-Weinberg law. The Hardy-Weinberg law, or Hardy-Weinberg principle, is used in studies of populations. It is used to derive genotypes from allele frequencies. So let me be very clear about what I mean by this. For all genes in our body, we have two copies, one that came from our mother and one that came from our father. And in many cases, in the cells in our body, those two genes are identical. So for example, we might have two copies of gene A in the cells of our body. However, some genes exist as more than one allele. For example, that gene could be found in one form, which we denote by a capital letter A, and another form, which we denote by a lowercase letter a. So for genes in which there is more than one allele, you can use the Hardy-Weinberg law to determine the frequency with which the different alleles occur together inside the cells of an individual. So in order to understand how this is used, you need to understand the terminology. So an allele is one of two or more alternative forms of the same gene. So a key point here is that this law is used to study single genes that have multiple forms. And an important distinction that you need to understand as a student is that this law is not used for different genes found at different loci on chromosomes or on different chromosomes. Different rules apply in those scenarios. This only applies to single genes that exist in multiple different alleles. To make this more clear, let's go through an example. Let's assume that a given gene has two possible alleles, which we'll denote as capital A and lowercase a. And let's suppose we know that the allele capital A is found in 40% of genes. We'll denote this 40% as the variable p, and we'll say it's equal to 0.4. If allele capital A is found in 40% of genes, then allele lowercase a must be found in the remainder of the genes, in other words, in 60%, because there are only two possible forms of this gene. We'll denote the frequency of allele little a as the letter q and say that's equal to 0.6. And this brings me to one of the first principles of the Hardy-Weinberg law, and that is that for a gene with only two alleles, p plus q must equal 1. In other words, if 40% of the alleles are found in form p, the remaining 60% must be found in form q, such that p plus q total 1. So what we're going to use the Hardy-Weinberg law to do now is to determine the frequency of the different combinations of genes to determine the different genotypes. All of us have two copies of gene A in the cells of our body. So what is the frequency of two capital A's being found together, two lowercase a's being found together, or a mixture of the two alleles being found together? On this slide, I've written our variables p and q in the top right corner. So once we know p and q, the Hardy-Weinberg law says that the frequency of two capital A's being found together is p squared. That would be 0.4 times 0.4, which is 0.16 or 16%. The frequency of a capital A and a lower a together is 2 times p times q. So that would be 2 times 0.4 times 0.6, which equals 0.48 or 48% of the time. And then finally, the frequency of two lowercase a alleles found together is q squared, and that's equal to 0.6 times 0.6 or 0.36, which is 36% of the time. And note that these three numbers all add up to 1.0. They have to, because these are the only three ways that the two genes can be found together, so they must add up to 100%. And that brings me to the second principle of the Hardy-Weinberg law, and that is this equation at the bottom left side of the screen, which says that p squared plus 2pq plus q squared is equal to 1. So what we've done here with this law is we've taken known frequencies of the two different alleles and we've used them to determine how likely it is that they will be found in all the different mixtures in which they can potentially exist in an individual. So let me just pause here and review what all the different variables in the Hardy-Weinberg law represent. At the top right of the screen, I've written the values we got in our example for p, q, and all the different combinations. So the first element of the Hardy-Weinberg law says that p plus q equals 1. In our example, p was 0.4. This means that 40% of genes in this population are big A. That's what the value of p represents. Q was equal to 0.6. This means that 60% of genes in the population are little a. We then use those values of p and q to calculate p squared, 2pq, and q squared. Let's talk about what those numbers mean. So p squared in our example was 0.16. This means that 16% of individuals in the population are big A, big A. 2pq was 0.48. This means that 48% of individuals in the population are heterozygotes. They have one big A and one little a. And then finally, q squared was 0.36. This means that 36% of individuals were little a, little a. Make sure you understand what all these values in this box represent intuitively, because you're often given a description of the value, and you need to figure out whether the question is describing the value of p, q, p squared, q squared, or 2pq. Now remember that at the beginning of this video, I said that the Hardy-Weinberg law is used in studies of populations. Well, if you want to apply the law to a particular population, that population has to meet certain assumptions that are inherent to the Hardy-Weinberg law. So let's talk about what those assumptions are now. First of all, the population has to be large. The law cannot be applied to small groups of people. In addition, there needs to be completely random mating. People can't be more likely to mate with the large A's and less likely to mate with the little a people, or this will make the law invalid. There can't be any gene mutations. Genes that are big A have to stay big A as they are passed down through generations, and genes that are little a have to stay little a as they are passed down through generations. There can be no migration in or out of the population. You can't have lots of big A's coming into the population and lots of little a's leaving. This will make the law invalid. And then finally, there can be no natural selection. If you have a selection pressure that makes it more likely that people will die if they carry the little a gene, then this will make the Hardy-Weinberg law invalid. If those assumptions are met, then for that population, the allele frequencies will not change from one generation to the next. In other words, if 40% of the genes are big A, then 40% of the genes will always be big A from one generation to the next for that population if all those assumptions are met. And if 60% of the genes are little a, then 60% of the genes will always be little a from one generation to the next. And thus you can apply the Hardy-Weinberg law in order to calculate the frequencies with which these two alleles will be combined. That is said to be a population that is in Hardy-Weinberg equilibrium. Now, in many cases, actual populations do not 100% meet all the assumptions of the Hardy-Weinberg law. However, if it is felt that a particular population is close enough to meeting them, then it will often be stated that they are close enough that you can apply the Hardy-Weinberg law to calculate gene frequencies. One of the places where the Hardy-Weinberg law is very useful is in autosomal recessive diseases. These are rare conditions, and in these diseases, the disease frequency is often easy to determine. For example, if we say that the disease version of the gene is lowercase a, and a person has to have 2 copies of lowercase a to develop disease, then it's fairly easy to figure out that 1 in 5,000 individuals have disease because they're sick and they are brought to medical attention. What's not known is the carrier frequency. In other words, how many people are walking around with 1 normal copy of the gene and 1 disease copy of the gene. And this can be determined by the Hardy-Weinberg law. And this is something you're often tested on. So let's go through an example of that now. So let's suppose that disease X is caused by a recessive gene. And let's suppose that we know that disease X occurs in 1 in 4,500 children. And we know this because these children are sick from disease X and they're brought to medical attention. Well, this means that we know the value of Q squared. Remember, Q squared is the frequency with which 2 disease alleles occur together. That is 1 out of 4,500 or 0.0002. From this, we can calculate Q. Q is the square root of 0.0002, and that works out to 0.015. What this means is that the frequency of the disease allele in this population is 1.5%. That's what the value of Q represents. Well, now we can easily calculate P because P plus Q equals 1. So P must equal 1 minus 0.015, and that works out to 0.985. This means that the frequency of the healthy allele in this population is 98.5%. We can now go ahead and use the Hardy-Weinberg law to calculate the carrier frequency. That's equal to 2 times P times Q. So that will be 2 times 0.985 times 0.015, which works out to 0.029, or roughly 3%. So what we've done here is used an observation about the frequency of sick children and used it to calculate the percentage of people in the population who are carriers. And we've done all this through the Hardy-Weinberg law. Now, one other point, for very rare diseases, P is often very close to 1. In this example on the screen here, P was 0.985, which is very close to 1. In these situations, you can often just assume P is 1 for simplicity and calculate the carrier frequency as 2 times Q instead of 2 times P times Q. In this example, if we had said the carrier frequency is 2 times Q, it would have been 2 times 0.015, and that works out to 3%, which is basically the number we got anyway. X-linked recessive diseases represent a special case where you can apply the Hardy-Weinberg law, but you have to understand a couple of tricks in doing so. So in order to understand this, let's imagine that we have an X-linked recessive disease such that individuals can carry either a normal X chromosome or a diseased X chromosome, which I will denote X sub D. In this situation, there are 2 potential male genotypes. Males can either be X sub DY or normal XY. There are, however, 3 potential female genotypes. Females can either have 2 normal X chromosomes, 2 diseased X chromosomes, or a mixture of the 2. The first trick to applying the Hardy-Weinberg law to X-linked disease is to consider the males and females as separate populations. If we first consider the males as one population, then among this group, P plus Q must equal 1. In other words, all males must have either the diseased X chromosome or the X chromosome. Once we accept this fact as true, then P must equal the frequency of healthy males. Any male with a healthy copy of the X chromosome must be a healthy male. Q must equal the frequency of diseased males. Any male with a diseased copy of the X chromosome must have disease. So, in other words, from just knowing the allele frequencies, we have also determined the frequencies of healthy and diseased individuals. We don't need to use Q squared, 2PQ, or P squared among the males. We just need to know the values of P and Q. Once you've done this, the second trick to applying the Hardy-Weinberg law to X-linked diseases is to recognize that males and females will have the same allele frequencies. So whatever P was for males must also be the P value for females. Whatever Q was for males must also be the Q value among females. Once we accept that, we can now determine the frequency of different genotypes among females by analyzing this population separately with the Hardy-Weinberg law. Among females only, P squared must be the frequency of healthy females with 2 normal X chromosomes. 2PQ is the frequency of carrier females with 1 diseased chromosome and 1 normal X chromosome. And finally, Q squared is the frequency of diseased females who have 2 copies of the abnormal X chromosome. And that concludes our module on the Hardy-Weinberg law.
Yo, I'll be back, man. Hello? ஏன் நேத்து போனீங்க இன்னைக்கு போயிருக்கனால? ஸ்டாலின் ஃபேமிலியா? அவன் சிஎம் இன்பன் அந்த பையன் தான் இன்பன். நீங்க பாத்தீங்களா? ஸ்டாலின் பாத்தீங்களா? அவரை ஹாப்பி பர்த்டே சொல்லுங்க. To you. Why? Deepa. எதுல? லவுன்ஸ்லியா? பீச்சா? எங்க சென்னையில தானே? ஓ, அந்த பீச்ல ஸ்டாண்டிங் இருக்காரா? ஓ, ஓகே. ஓகே, so then you அவந்தால வாக்கிங் போயிட்டு இருக்காரா? அங்க அவர் வாக்கிங் போயிட்டு இருந்தாரா? வாக்கிங் பரவாயில்லையே. அப்படியே அவரோட ஃபேமிலி ஃப்ரெண்ட்ஸ் ஆயிருக்கும்ல. அக்டோபர் நான் அக்டோபர் தான் இருப்பேன். ஆனா நீங்க அவனுக்கு ஆமா when you be there Chintona is also coming. Okay. நான் 10 ஓகே. நான் டாடி ரூம்ல இருக்கேன். 3 bedroom apartment அந்த தான் வராது. சோ எவ்வளவு அட்ஜஸ்ட் பண்ண முடியும்? நான் பேக் ப்ராப்ளம். யா ஜாலியா இருக்கும் எல்லாரும். யா யா ஒரு நாள் நான் எக்ஸாம் முடிச்சிட்டா யா கரெக்ட். Hello, I'm only 4 hours away. You can come here anytime. நீங்க வராததுக்கு நான் என்னது? திருச்சி எக்ஸ்டென்ஷன், டியூட்டி எக்ஸ்டென்ஷன். That means he has to work for longer time. When he is going to retire now. Oh. Oh. Okay. Okay. Super. ஆனா இதெல்லாம் நீங்க சும்மா சொல்றதுதான். அதான் சொல்லிருப்பீங்க ஆனா பண்ண மாட்டீங்க. பத்மாண்டே வராங்க. யா Okay. Yeah.
भारतीयार इंग्लिश this Basha
Hi, everybody, and welcome to our module on pedigrees. A pedigree is a representation of a family, and pedigrees are often used when analyzing or studying single gene disorders. A pedigree is a visual representation of how the gene is passed down through generations, and some members have disease and some members are carriers. And there are several typical patterns of genes that you should know for the USMLE Step 1 exam. These include autosomal recessive genes, autosomal dominant genes, and X-linked genes, and we'll go through all of these in this video now. Pedigrees use symbols to indicate whether a member of a family is male or female, whether that person is affected or unaffected by disease, and also to indicate whether individuals are married or are children of other members of the family. Unaffected males and females are indicated by either a white box or a white circle. Affected males and females are indicated by a line coming down from a particular marriage or union. Let's start by talking about pedigrees for all. Pedigrees for autosomal recessive conditions. In these disorders, there are usually two alleles for a gene. One allele is usually denoted by a capital letter, like capital letter A. That's the normal allele. And then there is a disease gene, which is usually denoted by a lowercase letter, like lowercase a in this example here. And only homozygotes for the disease gene, in other words, only individuals with two copies of the disease gene, will develop disease. So this is what a pedigree might look like for a family that has a gene for an autosomal recessive condition present in the family. First of all, note that there is an affected female up here at the top and an affected male down at the bottom. So this condition is affecting both males and females. Notice also that relatively few people have disease, and that's typical of recessive conditions. Because the gene is recessive, most people will either not have it or be carriers, but only a small number of people will inherit two copies of the disease gene and develop disease. Notice something else. Both of these individuals here do not have disease, but they are both carriers. The reason I can infer that from this pedigree is because they have a child who has disease. The only way a child is going to get disease is if they inherit one copy of the disease gene from each parent. Therefore, each of these parents must be carriers of the disease gene. So let's drill down on that in a little bit more detail. If both parents are carriers, in other words, if both parents are heterozygous and have one healthy gene and one disease gene, then the four possible combinations of genes that may occur in the offspring are shown in this 2x2 table on the slide. The mother has the potential to pass along a healthy gene or a disease gene to her offspring. The father similarly has a chance to pass along a healthy gene or a disease gene. This means that 25% or 1 in 4 of the offspring will have two disease genes and will therefore develop disease. 1 in 4 or 25% of the offspring will also inherit two healthy alleles and be completely normal and not be a carrier. And then 50% of the offspring, or 2 out of 4, will be a carrier for the disease and will have one healthy allele and one disease allele. And I've summarized these points in the slide. The child can have disease, only 1 in 4 chance of a child with disease. 2 of 4 children will be carriers, and 1 of 4 children will not be carriers. Another way of thinking about this is that the mother has a 50% chance of giving the disease gene A to a child. She has 2 genes, and for each one, there's a 50% chance she passes it on to her child. Similarly, the father has a 50% chance of giving the disease gene A to one of his children. Therefore, the chance that a child inherits 2 copies of the disease allele is 0.5 times 0.5, or 0.25, which is 25%, or 1 in 4, as we said before. And the reason it's helpful to think of it that way is because sometimes we don't know whether the mother or father are carriers, but we know the carrier frequency within a population. So let's suppose we know that the mother comes from a population where 1 in 50 individuals is a carrier of the disease gene. Let's suppose we also know that the father comes from another population where the incidence of carriers is 1 in 100. This means the chance that both the mother and father are carriers is 1 in 100 times 1 in 50, which is 1 in 5,000. Those two individuals then have a 25% chance of passing the disease on to their offspring. Therefore, the chance that a child of this marriage becomes affected is 1/4 of 1 in 5,000, which equals 1 out of 20,000. And you are sometimes given genetics problems where the risk of being a carrier is known like this, and you have to calculate them using the example I've shown on the screen here as a model. Let me make some more general points about autosomal recessive pedigrees. Some of these I mentioned earlier. In an autosomal recessive pedigree, males and females are typically affected equally. There are relatively few family members who have disease, and there are often many generations that go by without disease. And then finally, there's an increased risk of disease if a child is the result of consanguinity. Consanguinity is when parents are related and share common ancestors. If this happens, there's an increased risk that the two parents may both carry a recessive gene that runs in that family, and therefore there's an increased risk that a child will be born with that recessive condition. On this slide, I've listed just a few examples of autosomal recessive conditions. These include cystic fibrosis, sickle cell anemia, hemochromatosis, Wilson's disease, and there are many, many other examples of autosomal recessive diseases. Now let's talk about autosomal dominant disorders. In these disorders, the disease gene can dominate over a no disease gene. So usually there are two alleles for a gene in these disorders, just like an autosomal recessive. In this case, we'll call the capital A allele the disease allele because it's dominant, and we'll call the lowercase a allele the allele with no disease. And in autosomal dominant disorders, the disease gene is dominant. This means that both heterozygotes who have only one copy of the disease gene and homozygotes who have two copies, they both have disease. Shown on the screen is an example of an autosomal dominant pedigree. Note that males and females are affected equally. Note that when there is one parent with disease, there's a 50% chance that the offspring will have disease. For example, this woman here has disease and 50% of her offspring are sick. This man here has disease and 50% of his children have disease. Notice also that male-to-male transmission occurs in this pedigree. This is a male with disease, and he has passed along the gene to his son. This is an important fact because this lets us know this is not an X-linked disease. Males always pass a Y chromosome on to their son. They do not pass an X chromosome on to their sons. Therefore, if male-to-male transmission occurs in a pedigree, it means that the disorder cannot be X-linked. Shown on this slide are some examples of autosomal dominant conditions. These include familial hypercholesterolemia, Huntington's disease, Marfan syndrome, hereditary spherocytosis, achondroplasia, and there are many, many other examples. You should be aware that some autosomal dominant conditions demonstrate what's known as incomplete dominance, or they are said to be semi-dominant. This means that the heterozygote phenotype is different from the homozygote. It usually means that a heterozygote has a less severe form of the disease compared to a homozygote who has two disease genes. That person will have a more severe form of the condition. The classic example of a disorder with incomplete dominance is achondroplasia. This is an autosomal dominant disorder of bone growth. Heterozygotes for the disease gene will develop dwarfism. If you watch the television show Game of Thrones, the actor Peter Dinklage, who plays Tyrion Lannister, has achondroplasia, and that's why he has short stature. Homozygotes for the disease allele have a fatal condition and usually will not live beyond the first few months of life, and thus the disorder is said to be semi-dominant, meaning that the condition is different depending on whether a person is a heterozygote or homozygote. Another example is familial hypercholesterolemia. Heterozygotes have an elevated total cholesterol in the 350 to 550 range. Homozygotes have a higher total cholesterol in the 650 to 1000 range. Now let's talk about X-linked disorders. In these disorders, the disease gene is found on the X chromosome and is usually denoted as X sub D. These disorders will always affect males if they inherit an abnormal copy of the X chromosome. That's because males have only one X chromosome. Females can have one disease X chromosome and one normal X chromosome, and in this situation, the effect on the individual is variable. In X-linked recessive conditions, females are usually not affected, although we'll talk about some exceptions to this. In X-linked dominant conditions, females can be affected because the disease gene is dominant. So first, let's look at a pedigree for an X-linked recessive disorder. In this condition, all males with the disease gene have disease. They have no normal X chromosome to balance out the disease gene. Most females who have the disease gene are carriers and therefore will not be affected. If you look at this pedigree, only males are sick. This is typical of an X-linked recessive condition that runs through a family. In X-linked recessive disorders, there is no male-to-male transition. I mentioned this earlier, but all fathers pass a Y chromosome onto their sons. So note in this pedigree that even though this male individual here has disease, he has not passed it on directly to any of his children. The only way any of his offspring can get disease is if they get it indirectly through a female who is an offspring of an affected father. Sons of heterozygous mothers have a 50% chance of being affected. So for example, we know that this female right here must be carrying the disease gene because she has an affected son, and she is going to pass that diseased X chromosome on to 50% of her sons. And classic examples of X-linked recessive disorders are hemophilia A and B. These tend to run in families, and they tend to occur largely in males. In X-linked recessive disorders, females can very rarely develop disease. There are two ways this can happen. Usually it will only occur if a female is homozygous for the diseased X chromosome. This means her father must have the illness and her mother must be a carrier. The second way a female can potentially develop an X-linked recessive disorder is if she develops what's known as skewed lionization. So let's talk about that now. Lionization is a process that occurs in females and results in inactivation of one of the X chromosomes. Females have two copies of X chromosomes found in the cells of their body. One of those chromosomes in each cell will undergo a process called lionization by which it becomes inactive. Through this process, the X chromosome is condensed into a structure called heterochromatin, and the DNA in that X chromosome becomes methylated and therefore becomes inactive. In other words, all the genes on that one X chromosome become inactive. This will create something you can see under the microscope called a Barr body, which is found in the cells of females. It's a dark spot on the periphery of the nucleus. An example is shown here on the screen. That represents the inactive X chromosome in that particular cell. Lionization is a random process. Different X chromosomes become inactive in different cells in the body of a female. This occurs early in development at a stage when the embryo has less than 100 cells, and it results in X mosaicism in females. In other words, different cells in the body will have a different X chromosome that is working compared to others. This can potentially lead to symptoms in females of an X-linked recessive disorder if the process results in what's known as skewed lionization, where relatively more of the healthy X chromosomes become inactive and relatively greater numbers of the diseased X chromosomes are active, especially in the tissues where that X chromosome can cause disease. Now let's talk about the pedigree for an X-linked dominant disorder. These disorders can occur in both sexes, even though they are X-linked, because the diseased X chromosome is dominant. A special feature of these pedigrees is that every daughter of affected males has disease. This is because all daughters get an X chromosome from their father, and affected fathers must give their diseased X chromosome to their daughter. So for example, let's focus on this male individual here. He must have one diseased X chromosome and one normal Y chromosome. For him to have a female offspring, he must pass his X chromosome, which is diseased, onto his daughter, and therefore she will definitely develop disease. The pedigree of an X-linked dominant disorder can mimic an autosomal dominant pattern. However, a key difference is one that I mentioned earlier. In autosomal dominant disorders, there can be male-to-male transmission. In X-linked dominant disorders, there is no male-to-male transmission. That's because fathers always pass a Y chromosome on to their sons. So in this pedigree shown on the screen here, we have an affected male here. He has not passed it on to his son. He cannot do this because this is an X-linked disorder, and he is always going to pass a Y chromosome onto his male offspring. X-linked dominant disorders tend to be more severe among males because they do not have a second X chromosome that is normal. And the classic example of an X-linked dominant disorder is the Fragile X syndrome. I talk about this in more detail in the video on trinucleotide repeat disorders, but it is the second most common genetic cause of intellectual disability after Down syndrome. It is more severe in males. It often has features of autism, and children have a characteristic facial appearance like this boy shown on the screen here. They have a long, narrow face with large ears and a large jaw. Moving on, let's talk about mitochondrial genes and what a mitochondrial disorder can look like on a pedigree. You should be aware that each mitochondria found in our body contains some of its own DNA, which is called mtDNA, and this codes for mitochondrial proteins. If there is a mutation in the genes coding for mtDNA, the organs that are most affected will be the central nervous system and skeletal muscle because these rely heavily on aerobic metabolism. Mitochondrial genes display a unique genetic phenomenon known as heteroplasmy. It turns out there are multiple copies of mtDNA in each mitochondria, and in addition, there are multiple mitochondria found in each cell. If all of the mtDNA in a particular cell is normal or abnormal, that cell is said to display homoplasmy. The more common condition, however, is that cells display a mixture of normal and abnormal genes in the mitochondria, and this is called heteroplasmy. And because of heteroplasmy, mutant mitochondrial gene expression is highly variable. It depends on the amount of normal versus abnormal genes in each mitochondria, and also the number of mutant mitochondria in each cell or tissue. Shown on the screen, highly variable. It depends on the amount of normal versus abnormal genes in each mitochondria, and also the number of mutant mitochondria in each cell or tissue. Shown on the screen is an example of what a mitochondrial disorder pedigree could look like. In order to understand this pedigree, you need to appreciate that all of our mitochondrial DNA is inherited from our mothers. Sperm mitochondria is eliminated from embryos, and we only get genes for our mitochondria from our mothers. Therefore, if you look at this pedigree, you can see that a mother passes on the disease to her children, as in this example at the top. You can also see this mother here passing it on to her children. But note that affected fathers never pass it on to any of their children. That's because all of the mitochondrial genes come from the mother, who is not affected when there is an affected father that's a parent. Now, the expression of disease in the offspring of an affected mother is variable. If the mother is homoplasmic for the mutant DNA, then all children will have the mutation. However, if the mother is heteroplasmic, which is often the case, then the expression in the children can be variable. And because of this phenomena of heteroplasmy, the expression of mitochondrial disorders in offspring is highly variable. Mitochondrial disorders are very rare. One type that you're sometimes asked about on boards are the mitochondrial myopathies. This is a collection of rare disorders that all present with weakness, which is myopathy, also confusion because of central nervous system effects, and also lactic acidosis because you cannot perform oxidative phosphorylation if your mitochondria are abnormal. There's a wide range of clinical disease expression for all the reasons I mentioned on the last slide. And the classic hallmark of a mitochondrial myopathy is the presence of red, ragged fibers. This is seen on muscle biopsy with special stains. It's caused by compensatory proliferation of mitochondria in response to the mitochondria that are not working normally. As a result, there is an accumulation of mitochondria in muscle fibers that you can visualize under the microscope, and the mitochondria will appear bright red against a blue background with special stains. And this is an example of what red ragged fibers can look like on a muscle biopsy in someone with a mitochondrial myopathy. I'll finish this module by mentioning that many genetic disorders do not involve a single gene. Instead, they involve multiple genes and display what's known as polygenic inheritance. This is because many traits and diseases depend on multiple genes. For example, your height depends on many more than a single gene. Whether or not you develop heart disease or cancer also depends on the interplay of multiple different genes. So many of these disorders will run in families, but they will not follow one of the classic Mendelian patterns we've seen in this video, and that's because the disorders are polygenic. Here's a slide just to illustrate this point. Let's imagine 3 genes, A, B, and C, all influence the height of an individual, and we have 2 parents with a mixture of different alleles of A, B, and C who have a marriage, and they have a number of offspring. Some of the offspring will be very, very short because they will inherit only the lowercase alleles. Some of the offspring can be very, very tall because they will inherit only the uppercase alleles. And the rest of the offspring will be in the middle somewhere because they'll have a mixture of the 3 genes. And this is what you can see for any type of trait, including diseases that are polygenic. In addition to being polygenic, many traits or diseases are what's known as multifactorial. In these conditions, genes, lifestyle, and environment all conspire to create disease. And this is seen in many diseases like diabetes, coronary disease, and hypertension. It's not just a factor of what genes you inherit from your family. It's also got to do with your lifestyle, the dietary choices that you make, and the environment in which you live. And these are patterns of disease that are called multifactorial. And that concludes our video on pedigree. Hello, everyone, and welcome to our module on imprinting. Imprinting is an epigenetic phenomena. Epigenetics deals with variations in gene expression, not gene structure. So via imprinting, there can be an alteration in gene expression, and there can be differential expression of a gene depending on whether it came from the mother or the father. To understand this better, let's imagine that we have two copies of gene A found in the cells of a particular individual. One of those two copies came from the mother originally, and the other copy came from the father originally. Now, in most cells of our body, both copies of gene A can be expressed, assuming they're both functional and normal. However, for a small subset of genes, one of the two genes is said to be imprinted, and therefore it is not expressed. So, for example, if the maternal copy were not expressed, then only the father's copy of gene A would be expressed. And this phenomena is called imprinting. Imprinting occurs during gametogenesis before fertilization of the egg. Exactly how it occurs is incompletely understood, but genes are marked as being of parental or maternal origin. One of the ways they are often marked is by methylation of cytosine in DNA. At the bottom left side of the screen is the base cytosine, which you can find in nucleotides in our DNA. If it is methylated, meaning a methyl group is added, it becomes methylcytosine. And this is one of the ways genes can be modified sometimes in the process of imprinting. After conception, imprinting will control which gene is expressed. So in genes that are considered imprinted genes, only one allele is expressed. And this is in contrast, as I mentioned before, to non-imprinted genes in which both alleles are expressed. There are a small number of rare genetic syndromes that have been tied to the phenomena of imprinting. And the two classic disorders that involve imprinting are Prader-Willi syndrome and Angelman syndrome. What's interesting about these two disorders is that they both involve defects to the same segment of the same chromosome. However, you get a totally different condition depending on whether the maternal or paternal copy of the gene is abnormal. Both of these disorders involve chromosome 15, the section Q11 to Q13. So if you look at the bottom right side of the screen, chromosomes have a short arm, which is denoted with a P, and a long arm denoted Q. So the Prader-Willi syndrome and Angelman syndrome region of chromosome 15 involves the long arm, that's the Q arm, in the section from 11 to 13. If the paternal copy of this section of chromosome 15 is abnormal, the child will develop Prader-Willi syndrome. If the maternal copy is abnormal, the child will develop Angelman syndrome, and the differences are due to the phenomena of imprinting. To understand this better, at the bottom of this slide, I've drawn two copies of chromosome 15 in a particular individual. One copy comes from the father, that's this one here, and the other copy comes from the mother. On both of these genes, there is a section that contains PWS or Prader-Willi syndrome genes. However, these are normally expressed only on the paternal copy and not on the mother. And for that reason, I've written PWS. I've drawn two copies of chromosome 15 in a particular individual. One copy comes from the father, that's this one here, and the other copy comes from the mother. On both of these genes, there is a section that contains PWS or Prader-Willi syndrome genes. However, these are normally expressed only on the paternal copy and not on the mother. And for that reason, I've written PWS here on the left side by the father's copy. There is also a section of genes called UBE3A, and these are normally expressed only on the maternal copy and not normally expressed on the paternal copy. For this reason, I've written UBE3A over here on the right by the maternal copy of chromosome 15. In Prader-Willi syndrome, there is a loss of function of the paternal copy of the PWS genes. You can just remember that the P in Prader-Willi syndrome goes with the word paternal. And as I told you before, the maternal genes in this region of chromosome 15 are not normally expressed. So when you lose the paternal copies, you therefore have no functioning genes in the PWS region for that particular child. About 75% of cases of Prader-Willi syndrome are from deletion of the paternal gene, usually due to a sporadic mutation. Another quarter are due to maternal uniparental disomy. I discussed this process in the video on meiosis, but in uniparental disomy, two copies of the gene are inherited from one parent. In this case, two copies of the maternal gene are inherited and no copies of the paternal gene, and therefore both copies are inactivated. Prader-Willi syndrome is the most common syndromic cause of obesity. This is a picture of a child with Prader-Willi syndrome. At birth, one of the first things that's often noticed is that these children have hypotonia. They can have feeding problems, poor suck reflex. As they grow, they often have delayed milestones. They then develop hyperphagia and obesity. This begins in early childhood and persists through development. There's usually mild intellectual disability, and this is in contrast to Angelman syndrome, which we'll talk about in a minute, where there is severe intellectual disability. And then finally, these children often have hypogonadism and delayed puberty. Now let's talk about Angelman syndrome. In Angelman syndrome, there is an abnormal maternal chromosome 15 in the Q11 to Q13 region. This leads to lack of expression of UBE3A because, as I told you before, these genes are only expressed on the maternal copy, so when they are lost, you have no expression coming from the paternal copy. The majority of cases of Angelman syndrome are caused by deletions in the maternal copy of chromosome 15. A small percentage of cases are caused by uniparental disomy, and the reason this percentage is so small, much smaller than what is seen in Prader-Willi syndrome, is because you need paternal disomy in order to get Angelman syndrome. In other words, you need to inherit 2 copies of the gene from your father. Both genes will be inactive, and therefore you will develop Angelman syndrome from paternal disomy. In order for paternal disomy to occur, you need a meiotic non-disjunction event to occur in a male. And these events are much less common in males compared to females. In females, the process of meiosis is very drawn out, and therefore non-disjunction is more likely. The characteristic appearance of a child with Angelman syndrome is a child that exhibits frequent laughter and smiling. They are sometimes referred to as having a happy puppet appearance. 80% of children with this syndrome have a seizure disorder. They also often have ataxia and balance problems. And then finally, children with Angelman syndrome have a severe intellectual disability. And that concludes our module on imprinting.
Stream.